Are we in a position to manage these organisms? Still, our only solution that we have present is cut and burn, unfortunately. But the research that we're talking about, the discussion that is going to happen today, will tell you exactly our stark reality, what we are. Do you understand these organisms? Do you understand the other organisms which are involved? What kind of role they play? Are you in a position to manage them in the especially forest ecosystem? At present, we don't have much management, except there is one small silver lining is there. So these microorganisms, as Dr. Krishna Kumar mentioned, they take control of the entire cell of the plant which they infect, whether it's a plant cell or animal cell, and modify them. So that means they are working at molecular level. So if at all we want to use management technique for them, we have to also work at the molecular level. See, that is the intricacy that I wanted to emphasize today. So what I will do now is go through and then tell something about phytoplasm. Of course, there are specialists like Govindra, who is here. Later on, he will come. But to introduce these organisms, I'll start with that. Okay, what are phytoplasmas? No, earlier, as somebody said, they are called as, they were considered earlier as viruses. Later on, in 1967, Doi et al., Japanese workers, they found that they are not viruses, they are mycoplasma-like organisms because they are very close to mycoplasmas. So we started using the term mycoplasma. Then we know now that they are not mycoplasmas, but they are different. So we call them as phytoplasmas. But one thing very important, they are obligate parasitoids. They cannot be cultured because they need some system, plant system, to propagate themselves in the plant cell. So that's how they take the entire control of plant nucleus and then turn it around according to their advantage. Then they are polymorphic. Please. They are polymorphic, and they are gram-negative bacteria-like organs, but they are bacteria without cell wall. Then they multiply both in the, very peculiar thing about these is they multiply both in the plant cell as well as animal cell. That's vectors that they use. And as I told you, they modify the plant like anything. They can reduce the size of the plant, make it the leaf of the plant, make it more fleshy, more attractive to the vector species. They increase the nutrient content of their cell. They convert the flower parts to the vegetative parts, what we call phyllodes. Then the apical dominance is reduced, suppressed completely, so that the plant becomes branched, and then the internodal length will be reduced. So this actually completely changes the microenvironment of the plant, which is more useful for certain groups of insects. That we should understand, And these would be the mostly vectors. And nutrient will be changed, completely changed. So as a result, they become more attractive to the vectors, which transmit them, from one plant to the other. Then they also modify the entire, what they call, the chemicals around the plant. So that, and also they suppress the immune system of the plant because of herbivory. So all these actually they modify so that they can survive, attract the vectors, and then they can get transmitted. And also the plant, especially if the association between phytoplasma and the plant is very recent, then they can also cause death. If it is very old, they may not cause any death. Now, that is also a thing. Then how some affected plants may act as symptomless carriers. Maybe there may be a long association. I don't know. There may be a long association with the thing. And some plants may serve as dead-end hosts. This is very important. We have to identify because this will play a very important role in management of it. What are those dead-end hosts for each of the diseases that we are talking about? So dead-end host means the vectors cannot pick it up from that dead-end host and then transmit it. So that is a very important aspect of that. Then make the host more attractive. Of course, I mentioned about that. Then phytoplasmas are phloem bound, as I told, and they move through the sieve tubes. They have to go through. They are polymorphic, as I mentioned to you. They may be unevenly distributed. See, many times what happens when I discuss with the persons who are actually working with the phytoplasmas, they say, sir, we are using this particular vector, but it is proven as vector, but I am not getting any transmission. So these are the problems which are there. Because sometimes the phytoplasma may not be evenly distributed in the plant. It may be concentrated in a particular part of the plant. So roots are very poor. On the other hand, young leaves are also very poor sources. On the other hand, remember, in the case of viruses, young leaves are the best source. Whereas here it is not the young leaves, but it's the older leaves which are important. And also there may be seasonal variation, especially woody plants like forest plants. Seasonal variation may be there, especially deciduous plants are there. The phytoplasma will be going down into the stems or into the roots, and that's where it stays when the foliage falls off, So now we get into the transmission, because how phytoplasmas get transmitted from one plant to the other. Now, insects are the only or major vectors for transmission. In fact, they entirely depend on insects from plant to plant transmission, but there are other small ones, which maybe about 2 or 3%, they may rely on that. These are vegetative propagation. Use cuttings. If it is infected plant, you are going to. So that's why I think Dr. Krishna Kumar mentioned about it, that cutting should be avoided. That's one of the most important things. Similarly, bulbs, stolons. Sometimes there is suspect bacteria also can be transmitted, but I'm not sure whether it still stays there or not. Then there are also small number of phytoplasmas which are seed transmitted. So it may increase with our knowledge because you don't know much about it. Say, for example, tomato, phytoplasma which affect tomato, carnation, citrus, and also alfalfa. So we know that they are seed transmitted. Otherwise, they're largely dependent on insects. So among the insects, the phytoplasm depend only on one order of insects, that is Hemiptera. I'll tell you why at Hemiptera. So among the Hemiptera, it is the leafhoppers which are the major vectors. 81% of the known phytoplasm in the entire world are transmitted only by the leafhoppers. Okay, then remaining ones may be 7% or so by the psyllids, 6% by the delphacids, and then others, they are there. We'll talk about that a little bit. Vectors of phytoplasmas, as I told you, the Hemiptera, species of Hemiptera are the most important ones. These we call bugs, maybe shield bugs or aphids. All these come under Hemiptera. And in fact, all the vectors, majority of the vectors are in this group only, which have a very nice biological relationship with the pathogens. They are all in this group. Now, why Hemiptera, especially in this case? As I told you, the phytoplasma is phloem bound. It is not found everywhere. It is only in the phloem tissue you get it. So it needs a vector which can pick up, which can get into the phloem tissue, draw the sap along with the phytoplasma, and then introduce to a healthy plant and in this phloem tissue only. So this needs a highly specialized kind of mouthparts for this transmission. And this highly specialized kind of mouthparts are found only in Hemiptera. And this is the reason why you get majority of them are in this, okay, in this order. Okay. You can see, if you see the vector group, says nearly about more than 81% of that large blue that you see, they're all leafhoppers. Then you have 6 sets which are there, which are especially those which are transmitting the coconut wilt disease. There are 6 sets. Then you have psyllids which transmit the apple and then potato phytoplasmas. Then you have delphacids which transmit the rice phytoplasmas. And then you have durbids. One durbid is there which is also known to transmit coconut one. which is still suspected ones. So like that, and also there are two heteroptera. One has been, one is tinged, the lace-wing bug, and another one, as morning we mentioned, Heliomorpha halis. So these are again a question. But these 81% are not perfect. They're not confirmed vectors. I'd like to emphasize this. That I call them as putative vectors. They are suspected vectors. There may be about 50% of them which are very confirmed. We know that this is one. For example, we know for sure that Hishimonas fasciitis transmits little leaf of brinjal. For sure, we know that Oroceus albicinctus transmits Systemum phyllodi. For sure, we know that, okay, the grape Flavescens dori is transmitted by Scaphoideus titanus. So like that we know, but with the same confidence, I cannot tell that all these 81% can transmit. So again, this is a gray area where we have to work. And we have to know something about systematics of the leafhoppers, because it's very interesting that only a few groups of leafhoppers are capable of transmitting viruses, phytoplasmas, and also some of those bacterial diseases like especially Xylella. So why? See, classification of leafhoppers, in fact, is the largest family in Exoteriota, the grasshoppers and others you get. That's the largest. There are about 22,600 species known in the world. And we have in India about 2,250 species also in India. So it's a very large group. It's not a small group, it's a very large group with diverse habitats and diverse biology, very peculiar complex behavior they have. So of these, in fact, earlier, I have learned it as 50 subfamilies. In fact, my guru, Dr. Paul Oman, actually told, okay, there are 50 subfamilies of leafhoppers. But now, with advancement in the molecular techniques and also phylogenetic analysis using various techniques, the 50 subfamilies have been reorganized into 19 subfamilies. So there is a drastic change which is taking place. And even I have a lot of problems. Myself and my colleagues of my age have a lot of problems to cope up with this. We sometimes don't agree. We go on corresponding and then ask them, no, this is not right. This doesn't go into there. So like that. But let's not bother about it. What is important is now we have about 19 to 20 subfamilies of leafhoppers. And of them, very interestingly, nearly 75% of the vectors are in one subfamily called deltacephalini. Why this is the case? Deltacephalini is one of the groups which are highly evolved lineage. It's a highly evolved lineage. And the next one which is found is in Micropsini. And these are involved especially with acacia. In India, they are found on acacias. And most interestingly, here what happens, they're also fairly well evolved. So there is some kind of linkage between the evolution of the leafhoppers and the phytoplasm. As I told you, we have to look at, for the management, we have to look at the molecular level, not at the whole insect level or whole ecosystem level. It's molecular level also you have to look into. So these things would be of great importance when you are starting, planning the studies for management. So let's get into a little bit on the biology of these insects. Okay. Oh, is it okay? I have to see on this side? Could have told me that. It doesn't matter. We'll talk about a little bit on the biology. So most important is the host plants, because we have to know about the host plants of these. Very strangely, we can divide the host plants of leafhoppers into oviposition host plants, nymphal developmental host plants, and adult host plants, and to go on widening. The oviposition, they use a very, very small number of hosts. They're highly specific. The females are very, very specific for laying eggs. On the other hand, the nymphs, when they become a little bit wider, and the adults, they can feed on many, many unrelated plants. So this also has a very important, repercussion on the transmission. If they can acquire, phytoplasma in the nymphal stage, the adults can transfer this phytoplasma on several hosts without any problem. then there is often a problem in identifying the sexes in leafhoppers. Because when you have to rear them, I come up with, oh, sir, I don't know which is male and which is female. This is very important. So the female will, very interestingly, it will have a ovipositor, the one which is on your right-hand side is a female, one which is on the left-hand side is a male. So you have to check at the tip of the abdomen on the ventral side and see whether it has a ovipositor or not. And all leafhoppers lay their eggs in the plant tissue without any exception, absolutely no exception. So they make use of a very strongly sclerotized structure called as ovipositor, or what you call volvula. You can see on the left-hand side series of volvulae. So you can see some of them are smooth, some of them are teased, and they are used for that. They are using that and then inserting the eggs in the plant tissue. And the egg may take about, maybe if it is non-overwintering egg, it may take about 6 or 7 days, or even less if it is temperature is high. Otherwise, it can overwinter. Nymphs also can overwinter. And especially late instar nymphs can overwinter. The adults can also overwinter. So it depends on different species. They can overwinter. And this overwintering takes place in the temperate region, not so much in the tropical region where we are in. So tropical region leafhoppers are multi-volatile. You have a number of generations. So there should not be much of a problem here rearing insects, leafhoppers throughout the year without any problem. It is not the case with the temperate region where they have to have a controlled temperature, humidity, then they can break this dipause. Okay, there is a lot of variation in the host specificity, even among the same genus. For example, the green leafhopper, rice green leafhopper, Nephotodix. Nephotodix viruscens is highly specific. It only breeds on rice, nothing else. On the other hand, Nephotodix Nigropictus, Malayanus, Parvus, these will breed both on rice as well as the other grasses. So Hishimonas fasciitis, for example, and also Orosius, which are our most important vectors in India, they breed on several hosts, not one, on several hosts they breed. Macropsians again breed on a specific host. They are highly host specific. So this information becomes very important, There is also, we have noticed, especially when I visited Oman, I noticed that there is some population variation in the, of course, we know that there will be population variation in the transmission of the pathogens. But here it was very interesting that the person whom I was helping him to solve the problem of their, one disease, Phytoplasma disease, to identify the vector. He was maintaining the culture of Hishimonas. He was maintaining only on citrus. Then I asked him why you are not using the brinjal. He said, no, sir, it's not surviving. Then I was not sure. Then I asked him to put on brinjal. They died. So here is a condition where the host plants of different populations from different geographical regions may differ in their host plants preference. So you have to be very careful with this also. Now, feeding of leafhoppers is equally important because, as I told you, these pathogens that we are talking about are highly specific. They are confined to a particular region, tissue of the plant. For example, phytoplasmas are in phloem tissue. So only the phloem feeding leafhoppers can transmit it. So the leafhoppers can feed on mesophyll, they can feed on phloem tissue, they can feed on xylem tissue. So those which are feeding on phloem tissue are taking in sap, which is under high pressure, about 2 to 3 times atmospheric pressure, the phloem tissue sap is there. So they need not spend much, energy to suck the sap. So their face, their pump, they have a pump inside on the esophagus, we call it a pharyngeal pump. So after the mouth, you have a pre-cybarium, then cybarium. That's where the pump is there, which works just like any other pump, like your heart it works. So that pumps in. So you need little energy and little muscle. So that's why the faces of these leafhoppers which feed on phytoplasm, on phloem, they have a flat face, as you can see in this diagram, the first diagram. They have very, very flat face. On the other hand, those which feed on xylem vessels, xylem sap is under negative pressure, and they have to put in a lot of effort to take required quantity of fluid. And they have to have a very strong muscle to that. And then their face get bulged like a balloon. It becomes like a balloon to take in that sap, or pump in sap. That's why you can see in this particular second diagram, the lower diagram, you can see the ballooned face. So most of the deltacephalians, they are phloem feeders. Similarly, most of the leafhoppers are phloem feeders, whereas the cicadal lines are xylem feeders. It is important that xylem feeders are also equally important because they can transmit xylella fastidiosa, which is also, knocking at the doorsteps of India. It can enter into India anytime. Okay, this is something about their mouth parts. You can see, as I told you, they can enter into the tissue, and then with the least damage, you can see this is mouth part, this is leaf hopper, and you can you show it? So these things. You can see they cause very least damage, and so this is a stained one because when they feed, they leave a what you call they leave a protein band there. Salivary sheath, they call it a salivary sheath. It can be stained, and you can track where exactly the stylus go into that. Okay, you can go to the next one. So the mouth parts of these leaf hoppers have, sucking type of mouth parts. Okay.